Okay, so, oh, I'm up next. So I get to tell you now about some of our physiological work that's being assisted by genomics analysis, looking at the effects of the environment and the effects of host on tree defenses. And this is going to really set you up for the next two talks, which are by Nadir and by Maya, saying, well, this is what we know about what the tree did. How is that impacting on the beetle? Because the beetle is what moves, and it's the beetle populations that are important in terms of the risk models, although it's the pine that has the economic value. So pine trees have both constitutive, so preformed defenses, and induced defense responses to protect them from all sorts of invasion, including from mountain pine beetle. So this is the classic response to mountain pine beetle invasion by a pine, this copious production of resin or pitch, the classic term of being pitch out. So the beetle is poured in and the tree has responded through existing resources as well as new synthesis of these resins to try to not only drown out that beetle, physically push him out the door, drown him out, but also hopefully poison him and anything that happens beyond him, or her for that matter. And so you can see then, if you peel away the bark, that the tree has other additional chemical defenses that we can see. So this discoloration around this gallery that's been created by this beetle, those are deposition of secondary metabolites. So some of them are those famous terpenoids that you're going to hear about, others may have been phenolic compounds. And we can also induce a tree to respond by inoculating it with the fungal associates that ride along with this mountain pine beetle. So through various talks over the this week and last week, you've heard about fungi that sit in the mountain pine beetle gut and also in the head capsules. And so these fungi then play a role in actually creating conditions that will result in tree mortality. And so we can inoculate the trees with these fungal associates to induce the tree to produce a response, a defense response. And so you can see that discoloration here, deposition of secondary metabolites. You can also see that trees will ultimately respond by cells within that tree killing themselves off. This is a programmed cell death that these cells are undergoing in an effort to contain that invader. Okay, so in our field experiments, particularly in our growth chamber experiments, because we really want to be able to separate the signal from the noise, rather than inoculating trees with mountain pine beetle or relying on natural infestation where the certainty of attack is sort of always up for question and the level of attack can't be very well controlled. Doing the artificial inoculations is something approaching a wizard's art, doesn't always take. We've elected to use inoculation with the mountain pine beetle fungal associate for our experiments to ensure that we have a uniform response, we have a predicted course of response, and it's rather like using the glyphosate treatments that we heard about this morning as a proxy for mountain pine beetle to bring about a predictable death sequence to the tree. This is a way to initiate a defense response in the tree that partly mimics what the beetle does. Doesn't mimic the whole thing, and so Maya and Nadir will actually show that having the beetle present might certainly change things up a little bit, but this gives us a picture. So here are our fungal associates. They're really pretty up close. I have to say, I'm not a mycologist, but I do think these are really lovely. And so they are actually responsible for that sapwood discoloration that we see, the blue stain fungi, in trees that have been attacked some years previous. And what they're going to do is they're going to grow into the sapwood, and should they manage to grow sufficiently, they will block those conducting elements of the sapwood, so blocking the ability for water to move up that stem. They're going to be able to break down cell walls in these cells to actually be able to attract those nutrients, but most importantly, they can get a hold of materials from these rays. And these rays are living. These guys, these conducting elements, they're dead. These rays, they're living. They contain important nutrients. Those then become available to whatever is burrowed in here with those fungi, i.e. the mountain pine beetle. So, mountain pine beetle overcome pine defenses through mass attack. If one single beetle attacks the tree is likely to be able to win that battle. The tree is likely to be able to produce enough defense to pitch that beetle out. If a few beetles attack that tree's still going to probably win. But as those numbers of beetles that attack the tree increase, the beetles then will be able to overcome the defenses that the tree is able to garner to protect itself. So these are the classic curves from Raffa and Berryman in the 1970s and 80s. So if we look at increasing numbers of mountain pine beetle that attack a tree versus the outcome of mountain pine beetle reproduction, we see that in the early phases of an epidemic when populations are a little bit lower, that an undefended tree is going to result in successful attack and successful reproduction, but a well-defended tree is going to be less than this ideal in terms of the mountain pine beetle perspective. And so this part of the curve, where attack densities are below a certain threshold, is affected by the host, that is the genetics of the host, as well as the environment that the host is in. Beyond a certain point, the trees are basically toast. There's enough mass there that when the tree gets attacked, there simply isn't enough defensive resource available for the tree to mount a successful attack, and the beetles will win. But down here, at the lower attack densities, the tree has a chance to win. And a well-defended tree has a better chance to win compared to a tree with less defensive capacity. So one of the classic findings of Berryman is that stressed trees are more of a favored target in those lower mountain pine beetle densities. As those population levels then rise, healthy trees become a more favored target or victim. And so these are borne out from the studies that, say, Kathy Bleicher has done in BC, that in those low population levels, it's the stressed trees, it's the older trees, the weaker trees that tend to succumb to attack, whereas at the higher epidemic levels, it's those large healthy trees that are attacked. And large healthy trees, that's the carrot cake that mountain pine beetle wants. That's where all the good feeding is to be had. The weaker trees, it's assumed that the resources that are available, the phloem thickness available, the water content, is less conducive to having a large, vigorous, bright family. So we know from some recent work that Alvin Yanchuk's group has done that there is a moderate genetics component to resistance in lodgepole pine. This is work that was done just outside of Prince George at the Red Rock Nursery in a full partial diallel provenance study that where the epidemic came and went, and so they saw moderate heritability for resistance to initial attack. But given the genetic architecture of this trait, this is not a single gene resistance. It's not even a small number of genes that are likely contributing to this resistance, which I should stress is moderate, but it is clearly controlled by many, many genes, each with very small effects. But it does say that there is a genetic contribution to the tree's resistance and the ability of the tree to defend itself. So bearing this in mind then, what we've concentrated on in our aspect of the project is asking whether defenses between lodgepole pine and jack pine differ, based on the idea that if there are differences between defenses that are put forth by different lodgepole pine with different genetic backgrounds, one might expect that there would be differences between lodgepole and jack pine, particularly because lodgepole pine is the co-evolved host, so it's co-evolved with mountain pine beetle. It's presumed that in the arms race between mountain pine beetle and lodgepole pine, that lodgepole pine will have evolved defenses to deal with this particular bark beetle, whereas jack pine is considered the naive host, it's the new host, and so there is no co-evolutionary relationship there. If we build a model of what defenses might have looked like and might have evolved, one might predict then that jack pine will not have a co-evolved defense, and so it's relying on its generic defense against bark beetles when it's attacked by mountain pine beetle. So here in Alberta, we've heard lots this morning about how this side of the rocks is a drier climate than many parts of BC, and so to fill that knowledge gap for modeling purposes, we decided to concentrate on the effect of water moisture availability or water deficit on defense responses. So there's been a little bit of work that has been done in southern pine beetle and the effects of water availability on defenses and southern pine beetle, not so much done on mountain pine beetle. So we chose to use two different approaches. Growth chamber studies, where we could have ample repetition, we could do a full time course with seedlings, we could have genetic uniformity, although they're not the same genetic background completely, they could come from the same provenance. And we could really adequately control water moisture in our pot conditions. But of course, mountain pine beetle don't attack seedlings. So we've taken genomics outside, and we've also done mature tree studies with hybrids, with lodgepole and with jack pine. The hybrid experiment was carried out in 2009 near Whiteport. Lodgepole and jack pine study for our study was carried out at Smoky Lake at the Provincial Tree Improvement Center there. We used native stands of even-aged jack pine and planted stands of lodgepole pine. And this is really just because the logistics of carrying out these gene expression studies outside are not trivial. And to have two sites really, really far apart, one for lodgepole and one for jack pine, not only would present us with logistics that were beyond the budget that we had, we would also have to contend with site variation that would affect our ability to make inferences between the treatments. So we had two treatments, well-watered or water deficit treatment, which was measured by TDR, which we heard about this morning, time domain reflectometry. We had control seedlings, wounded seedlings, and wounded plus inoculation with one of the mountain pine beetle fungal associates. You might hear about it, Grossmannia clavigera. It doesn't really matter what it's called, we'll just call it the fungal associates. And so in our field experiments, just due to the logistics again and the number of trees that we could get in a site, we used the control in the wound and inoculation treatments, but we didn't have a wound-only treatment. So pines are conservative water users. They typically show what we call isohydrant behavior, so using somatal opening and closing to control water movement and water loss. They would sooner conserve water and risk cavitation than they would spend that water and continue growing. So like many of the American pines that have been studied, lodgepole pine and jack pine also show that behavior in response to water deficit. So we see, even under the relatively mild degree of water deficit that was applied in this particular study, both lodgepole pine and jack pine closed their stomata in response to water deficit. in the very short time frames of our experiment, and we have actually continued this out to 70 days in particular experiments, we didn't see an effect of the fungal growth on more excessively on stomatal conductance or the ability of the stomata to open and close. But one would expect that to happen in the later stages of mortality. What we're really interested in is seeing how the tree is responding to that initial barrage from the mountain pine beetle and their fungi, rather than the consequences of colonization. So that's why we focused on these early time points. So because the stomata have closed, so there is less air exchange between the tree and the inside of the tree and the environment, then we get a reduction in photosynthesis. So these trees then have reduced carbon gain under water deficit conditions. And I want to stress again that this is not a very severe water deficit condition that we've imposed on these trees. Both lodgepole pine and jack pine, in all of the experiments we've now run 6 independent experiments in space and time, with the exception of one provenance of jack pine seedlings, they all show this behavior. So we looked at this lesion development, or this reactive zone, as a measure of the tree's defense response to invasion by this mountain pine beetle fungal associate. So this is our lesion here. And so in our seedlings, what we see is that lodgepole pine mounts a fairly rapid response to having been invaded by this fungus, even as early as 7 days post-inoculation under well-water conditions. Under water deficit conditions, we see that that response is delayed. So they may achieve the same lesion length, but it's delayed by a fair number of days. And so what this is telling us, if we go back to classic plant pathology literature, is that under well-water condition, lodgepole is able to mount a very rapid response, and that's a containment response. One would expect then that that lesion will effectively prevent that fungus from escaping and colonizing outside of that defense zone. Whereas the longer it takes for the tree to recognize that it's being attacked and mount that defense response, the more time then that fungus has to penetrate that tissue and establish and colonize that tissue and escape that defense response. And so under water deficit conditions, we have that delayed response. One might expect then that the fungus and the beetle that's with it would win more times than not compared to under well-water conditions. If we look at the jack pine responses, what we see is that it takes longer under both well-water and water deficit conditions to mount that defense response in the seedlings. And that, in fact, under water deficit conditions, we have greater lesion development at 56 days post-inoculation, which was quite interesting. So these trees, being a little bit stressed, maybe tip them over the edge to develop that lesion. Mature trees show much the same trend. This is data that I'm taking on lesion length that occurred over one growing season. This is what we were able to do. But of course, it's really the next growing season where we see the consequence of having been attacked. And so we would expect that although we see the same trends within one growing season, if we were to continue this through to a second growing season, that we would see a pattern like this where the water deficit trees would probably show a greater lesion length in the second growing season, whereas we might see a little bit more of a decline under well-water conditions. But regardless, in those early days after inoculation, under well-water conditions, we have a rapid response. We have an attenuated response under the water deficit conditions. And again, in jack pine and water deficit conditions, showing more of a response, though it's not significant, those later days. So we have started our gene expression analysis. We've looked at a total of 20 different genes. We've also, I'll show you in the next slide, started our genomics analysis. And what's very interesting is that what we see is under water deficit conditions that some genes that are involved in defense response show an increase in expression under the constitutive response conditions. So they've been turned up as the plant doesn't know it's going to be attacked, but its basic defense against whatever happens to be out there has been turned up in response to being water stressed. So for some genes we see that, for example, this chitinase, which is a nitrogen-based response, we see under water deficit an increase in expression of a constitutive response. In this 3-carrying synthase gene, which is part of that pitch synthesis, we see again the increased response under water deficit, so increased defense at the constitutive level. But then that response is attenuated once the tree actually is attacked. It doesn't increase over and above what it pretty much did already as a constitutive response. But what we do see is that other genes belonging to the same families actually show a much attenuated response after the tree's been attacked. So the induced response to being attacked just isn't there for some of these genes. So what that means is that, how we interpret these data anyways, is that mild drought response or mild drought stress is causing the tree to enhance its overall stress response package that is presenting to the environment. But there's not a lot of extra resources in the bank. So if the tree does get attacked, there's not a lot of gas left in the tank to mount the induced response. That's the pattern we get out of 20 genes. I show you 4 here. So now what we're doing is looking at those 25,000 genes across all of those different treatments and combinations of the time course, which is, as we're looking at wood and phloem, or the bark, that's about 350 different microarrays, so that's like a lot of data. So hopefully at some point we'll be able to put together all of that gene expression data to tell you a more concerted picture of what's happening at the constitutive levels, the preformed responses versus the induced responses. Overall, is a snapshot picture, an accurate representation of what's going on globally with gene expression. It also points us to the genes that change in gene expression with response to an environmental stress like drought, and also in response to mountain pine beetle attack. We can identify the genes that are potentially important to the tree in that response, and then we can ask, is there genetic variation that occurs at the site of those genes between individuals, and can we detect pattern on the landscape? So Kathy will talk about that after we hear from Nadir and Maya on. We see different, we'll get to questions just a sec, but they're going to tell us, does this matter to the beetle? If the tree has a different defense, does the beetle care?